Okay, hello everyone. I'm Mobilas Banerjee and you are watching Achievers Biology. Please do like and subscribe the channel and the video. Uh, we will discuss in both English and regional language in Bengali for some students. Today we will discuss on the blood grouping. A complete blood group type would describe a full set of 30 substances on the surface of red blood cell. On the basis of different substances antigens present on the surface of red blood cell, we will divide blood group into different subtypes. According to the 30 blood group system are being discovered on the basis of 310 approximately types of blood grouping antigen present on the surface. There's a major are ABO, RH, but there are also some present, that is MNS, Duffy, etc. etc. So if we discuss about the ABO blood grouping, a series of tests reported by Karl Landsteiner in 1900 when he was doing a blood transfusion. So what is the importance of ABO blood grouping? The first ABO blood grouping was discovered for the base of blood transfusion. ABO blood grouping is very important to avoid the acute hemolytic transfusion reaction. রক্তের সঞ্চালন ঘটিত কারণে কোনো সময় যদি রক্ত প্রদান করতে হয় তো সেক্ষেত্রে ব্লাডের তঞ্চন রোধ করার জন্য আমরা ABO blood grouping খুবই গুরুত্বপূর্ণ. Now ABO blood grouping is based on presence of A antigen and B antigen on the surface, of which all these two antigens are discovered from are produced from H antigen, which is the precursor antigen. Now genes of A and B codes for the corresponding antigen on the red surface cell, but gene O are amorph. It does not produce any antigen on the red cell surface. According to all this, group A blood cell containing only A antigen, group B blood cell containing B antigen, group AB blood cell containing A and B both antigen on the surface, whereas the group O red blood cell contain only H antigen, so neither A nor B. A blood cell আমরা A antigen পাবো, B blood cell ক্ষেত্রে B antigen, যদি AB blood grouping হয় তাহলে সেক্ষেত্রে AB both antigen পাবো এবং O blood grouping ক্ষেত্রে AB কোনো অ্যান্টিজেনই পাবো না, শুধু H অ্যান্টিজেন পাবো. Now we have to discuss about the antiseras. So what is antisera? Now we all know about the antigen and antibody. In case of antigen, it is a protein like substances which is responsible for the immunogenicity in the বডিতে ইমিউনিটি জেনারেট করার জন্য আমাদের অ্যান্টিজেন প্রয়োজন. এর এগেনস্টে আমাদের বডিতে অ্যান্টিবডি প্রডিউস হয়. on the against of antigen, body produces antibody. So if body has antigens on the red cell surface, there will be definitely antibodies corresponding to it on the serum, the blood. So in case of antisera, if we consider group A antisera having anti-B antibody corresponding to A antigen on the red cell surface, group B blood grouping having anti-A antibody on the serum which having B antigen on the red cell surface, group O individuals having anti-A and anti-B antibodies on the serum in corresponding no antigen on the cell surface, group AB individuals having neither anti-A nor anti-B because its blood cells having A and B both antigens on the surface. So on the basis, when we are doing the blood grouping test, we are using the antisera substances as a reagent. So when it reacts with the cells, it will coagulate and we discover the blood group of the individual. If the individual is A blood group, his cell will give coagulation on the basis of anti-A substance. If it's B blood group, then it definitely gives coagulation on anti-B antisera. If it's AB blood group, then it will give coagulation on the basis of both A and B antisera. If it's O blood group, it's neither give coagulation in anti-A nor in anti-B. Now what is the structure of this A antigen and B antigen? In case of A antigen, is a substance N-acetyl galactosamine which is present on the red cell surface, and in case of B antigen, that is the D-galactose. Now the precursor chain present is H substance, which is both present in A blood group as well as B blood group. In case of H antigen, there is neither present of D-galactose nor present of N-acetyl galactosamine. So the H substance precursor chain, it will present only in case of H antigen. In most individuals, A, B and H antigen, in addition to being present in the RBC, can also be detected in secretions of blood groups such as sweat and saliva. These persons are called secretors. People who lack blood group antigen in secretions are called the non-secretors. Now these blood group antigens look like this. ABO blood group antigens are being a little more extended from the blood grouping system, whereas the RH blood grouping antigens are submerged. Now A blood group have some subgroups. It's mainly A1 and A2, and the name discovered from Dolichos biflorus. 80% of individuals of A and AB individuals having A1 blood group. There are other subgroups present, among which A3, AX, AM, AN, etc. etc. A3 red blood cells cause mixed field agglutination when polyclonal anti-A and anti-AB is used for the detection. Mixed field agglutination appear as small agglutinates with a background of ABO. Now what's the difference? 1 to 8% of A2 individuals produce anti-A1 in the serum, whereas 25% of A2 individuals produce anti-A1. If we discuss about the B subgroups, there are also some subgroups in B. The B subgroups are differentiated by types of reaction with the anti-B and anti-AB, and also with the anti-H. These are the 3 B subgroups. H antigen is the precursor antigen, so it will present both cases of all the blood groups. On the basis of this, there is a greater number of H antigen as well as the least amount of H antigen we can detect here. So thank you everyone for watching this video. Bye.